Good morning, everyone. Do you mind? If you can turn off your audio, please? Thank you. Let's wait for another minute so we can get started.
Good morning, Professor.
Good morning. Thank you. Okay, so let's get started. Okay, all right. So last time we have learned the 3-point test cross. So this is very important. I hope everyone can understand what is going on in the 3-point test cross. So basically, we do a test cross based on tri-hybrid, right? So we have, for example, A plus B plus and C plus, and we have another chromosome is A, B, C. So this is the tri-hybrid. And we test across this one using the homozygous recessive. And then we can get a bunch of progeny. So based on the phenotypes of those progeny, so we can derive the map of the 3 genes, right? First, so this is different from what we have learned before the midterm 1. So before midterm 1, we were talking about the genes that are unlinked, which means those genes are located on different chromosomes. So now we focus on genes which are linked, which means the genes are on the same chromosome, but we do not know how these genes are arranged along this chromosome. That's why we're going to do this test cross, the 3-point test cross. And based on the information we observed from the progeny, we can derive the map. All right. So, for example, this is the map. We need to, for example, in tomorrow's quiz, and also in the coming in the next midterm. So I'm going to give you questions like this. So I'm going to give you a 3-point test cross, and I'm going to show you the data for the progeny, and you need to use that data to derive the map like this. So first, you need to figure out the order, right? So which gene is in the middle, and then based on the data, we will be able to calculate the distance, genetic distance between these 2 genes and these 2 genes, and also these 2 genes. And using this map distance, you can easily construct the genetic map for these 3 genes. So this is what we have learned last time. All right. So now the question is, all right, are this crossover? So for example, this is the same example. We have 3 genes, and using the progeny data, we successfully derived the map like this. That the CT gene is standing in the middle, right? Okay. Now we want to know. Suppose this is the region 1. So if we say that the region between gene V and the gene CT is called the region 1, and this is the region 2, which is the region between gene CT and the gene CV. All right. So suppose there's a crossover occurring in the region 1, and maybe there's another crossover occurring in region 2. So we know this is called the double crossover, right? So if this is true, we want to know if the crossing over in region 1 is independent of the crossover in the region 2. So this is the question we want to address now. Are crossing over independent, right? Okay, so how do we answer this question? How can we answer this question? So here, to answer this question, we're gonna apply some statistical principles here. So I believe Lei has already taught this in the discussion session, and I also mentioned some statistical principles before. So here we're going to use the multiplication rule in statistics. Suppose let's say, oops, where's my, okay. So suppose we say the crossing over in region 1 is a event. This is one event. And we say if a crossover occurring in the region 2 is another event. If these two events are independent, then we can calculate the joint probability for these two events, right? Joint probability that two events both occur will be this. So remember, what is the genetic distance between these two genes? So this genetic distance is defined as the recombination frequency, right? So what's the chance that you're going to see a recombination occurring between these two genes? So that's the probability for event 1, and that's the probability for event 2. So if we assume these two events are independent, so we can simply use this multiplication rule to calculate the probability that the joint probability that these two events occur simultaneously, right? At the same time, right? So, oops. Okay. So this is the expected frequency of double crossing over, which is the probability of crossing over in the region 1 multiplied by the probability of the crossing over at region 2. So since this genetic distance is the probability, so we can simply multiply these two numbers, get this. All right, so this 0.008 is the expected frequency of double crossover in this entire region. All right, now if we have the expected frequency of double crossing over, then if we knew the total number of the progeny from this test cross is this many, and then we can easily calculate the expected number of the expected number of the progeny having double crossover should be this number multiplied the total number of the progeny, which is 12. All right. So how do we understand this? This 12 means if the crossover in region 1 is totally independent of the crossover in region 2, then we are supposed to see this many progeny carrying a double crossover. All right. So, however, if you go back to the problem, so you're going to see the observed double crossover is only 8. So this number is smaller than this one, it's less than this one, which means what? So because the 12 is the expected progeny with double crossing over only if these two regions are independent, right? So the crossing over in these two regions are independent. However, the observed number is significantly smaller than this number, which means our assumption is not right, which means these two regions are not independent. So I hope you can follow my logic here. All right, so if this makes sense to you, so then we can make a conclusion here. So so in this case, the crossovers are not independent, which means if we look at the entire region of this, so the map of the three genes, so if there's one crossover occurs everywhere, anywhere, so that one crossover will create traffic that can inhibit the second crossover in this region, in this map. Does that make sense? All right, yeah, I hope, yeah, this is clear. And this is very important. I'm going to show you an example here. Before the examples are still important concepts. So one important concept here is the interference
Professor,
interference coefficient. Yes, you have a question?
I'm just wondering, so the 12 value, that just means that that's the like frequency of double crossovers in any two segments of that gene. It doesn't have to be like the same segment.
So in this case, that's a good question. So in this case, we purposely separated the entire map into two segments, 1 and 2. Because if you use a random segment, then you can never calculate these numbers, right? Because we just take advantage of this map. So we know that the map is like this. And for this two definitively defined region, we can calculate the probability for this region and this region. If we do not use this segment, let's say we truncate the map into this region and this region, but based on the progeny data, we cannot calculate the probability of crossing over in this region and this region, right?
And each of those regions, like the split is like one gene on that chromosome?
Yes, so basically, yeah, this is the case we use for calculation for this problem, yes.
Okay, thank you.
Does that make sense? Great. Oops. Okay, so. Okay, so now let's introduce a very important concept here. So this new concept, yeah. So you still remember, right? So the last question I gave you in the midterm is a bunch of like important concepts. So this one is also very important. So hopefully, you take notes, and so this might appear in your next midterm. All right. So interference coefficient. So we use I to represent this. And so this I interference coefficient is a measure of the independence of crossovers in a chromosomal region. So this is a chromosomal region, right? This region represented the map of the 3 genes. So if we want to calculate the I interference coefficient in this region, so this is how we do this. So the I is actually, so this interference coefficient equals to 1 minus C. So C is another concept that is associated with I. So the C is called the coefficient of coincidence. All right. So, and C is defined as is defined as a ratio of what? The ratio of observed double crossover progeny. So this is from our data, right? So if I give you a table containing the information about the progeny from the 3-point test cross, so you can easily figure out this number. The challenge is, like, if I give you a problem like that, you have to calculate the number in the denominator, which is the expected number of the double crossover progeny. So in this example, this number is 12. So I hope everyone understand how we got this number. I'm going to show you later. So I'm going to use an example to show you how we calculate this number again. All right. So in this case, the C equals to this, and then it's easy. So the I, so the interference coefficient of interest is 1 minus this equals to this. All right. So as you can see, so when we so I could take different values. So if the interference coefficient equals to 1, which means what? Which means there's no double crossover at all. So if there's one crossover, this one crossover will completely prevent other crossing over occurring in the same region. So in this case, we're going to say, so I equals 1 means a complete interference. So you will never see double crossover in this map, in this chromosomal region. All right. Another extreme scenario is if I equals to 0. that's easy, right? That means like there's no interference at all. So if there's a one crossover occurring there, so it doesn't matter. So another one could also occur here. It will not affect any additional crossover here. So that means like these two regions, in this case, these two regions are independent. All right. And in most of the situation, actually, I take some value between 0 and 1, which means there's a interference, but there's a different level of the interference. All right. And in this case, you're going to see the expected double crossover should be greater than the observed one. All right. So those are the most common situation, and there's some very special situation here. So I could be less than 0 is a negative value, which means what? Which means the observed double crossover is greater than the expected double crossover progeny. So what is that? What is that? So why this could happen like this? So which means, so the biological reasoning is, so if there's one crossover occurring in this region, it will stimulate some other additional crossover occurring in the same region. So this makes sense. This is not common situation, but this makes sense because so I hope you learned biology before. So on chromosome, there's some region on any chromosome. There are some very special region called the recombination hotspot. Hotspot means like the crossing over tends to be occur there very frequently. So in that region, the recombination hotspot, this could happen. So the I could be less than 0, which means like if there's one crossover, that crossover stimulates additional crossover in that region. I hope this makes sense. Okay. All right. Let's look at an example here. All right. So this is a solved problem, I think. Again, so some students have asked me, so before the midterm, so how can we like well prepare for the midterm? So this is one way. So because on the syllabus, I gave you the reading assignment, so that's important because reading textbook will help you understand the concepts we covered in the lectures. And also, I gave you the side problems. So those are not required homework, so it's up to you. But I strongly recommend that you do those practice problems because that can help you understand the concepts. And also, sometimes I can give you very similar problem sets. So if you do the practice problems, that can help you be successful in a midterm and a quiz. So this is one example. So solve the problem. So in this problem, so I'm going to give you a 3-point test cross. Okay. So we have no problem. We should have no problem with these concepts, right? So here we knew these 3 genes are on the same chromosome. They are linked, but we do not know their arrangement on the chromosome. So we need to derive the genetic map for these 3 genes. And this is the classical way we can do. We do the 3-point test cross, right? So the first parent here is the trihybrid, right? So we have 3 genes, and each one has a heterozygous locus. And we test cross this one with the tester, which is the triple homozygous recessive. And this is the data. So in the test, so you're going to see, I'm going to give you data like this. And so those are the data. So immediately you can identify which are the which 2 categories represented the parameter type, right? Because these 2 big numbers, greatest numbers. should be the parental type. And look at these two categories with the smallest number. So that should be the double crossover progeny, because crossover is a very rare situation. That's why many of the progeny will show the parental type. And the least category will represent the double crossover because of the rare situation. All right. Okay, now when you compare this double crossover progeny with this parental type, so the first two are identical. Only this one has been changed. So this information can give us what conclusion. So because if you go back to the previous slides, you're gonna see if you compare the parental type with the double crossover progeny, the only changed locus is the gene in the middle, right? So based on this information, you can immediately say the gene CN is standing in the middle. So B should be here and the WX should be here. So, or you can exchange the position of B and WX, that's fine. But as long as you figure out the CN is in the middle, yeah, that's correct. That's the correct map. So if we do not worry about the central mirror in the mapping, so this is, so either way is correct. So I don't worry. I'm not worried about how you put the genes on two ends, but the only important thing is like you need to figure out which gene is in the middle. So that's the key point. All right. So okay. So now, so we have this draft map, and then what you need to do is like, so if you look at each pair of the two genes, each gene pairs, you're gonna calculate the genetic map, right? So first you need to figure out for this gene pair, what are the recombinant, and you figure out the recombinant category and calculate the sum of the recombinant progeny divided by the total number of the progeny. So that's the recombination frequency between these two genes, right? And then you can do this for all the gene pairs here to get this. And okay, now you have three numbers. Don't worry about the biggest number, because we're not gonna use that number. We're gonna use the two smaller numbers and put these two numbers in the map. All right, so in the test or in tomorrow's quiz, so if you can derive this map, then you're gonna get the credits points, all the points. All right, that's very easy, right? All right, okay, now let's continue to look at this. All right, so if I ask you to calculate, if I ask you to let me know if in this region, in this chromosomal region, the crossing over are independent or not, so which means you need to calculate the interference coefficients based on this number, right? So that's also easy, right? Because okay, so suppose if the region 1 and the region 2 are independent, then the expected frequency of double crossing over should be what? Should be the multiplication of these two numbers, right? These two numbers here. All right, that's the expected frequency for the double crossing over. And then if you multiply this number by the total number of the progeny, that's the expected number of progeny carrying double crossover. All right, and in the data, so you already have the observed the number of the double crossover, which is 11. That you just simply use this formula to calculate the interference coefficient, which is this. So this is a number, positive number, which is less than 1, which means there's a certain level of the interference between within this chromosomal region. So does that make sense? Any questions? So I'm gonna pause here so in case you have questions.
I had a question.
Mm-hmm.
For the previous slide, I didn't really, so could you explain again how you knew that the CN was the one in the middle, and then like how so the numbers are what again?
Yeah, sure. So I think maybe we can go to this slide. So this is the first time we introduced 3-point test cross, right? All right.
Mm-hmm.
Okay, so if, let's see, this is the parental type, right?
Mm-hmm.
Okay, now if there's a crossover occurring between A and B, then we're gonna get this and this. So if you compare this one with the parental type, only A has been changed, right?
Yeah.
The same thing on this side, same. Okay, if there's a crossing over between B and C, so if you compare this progeny with the parental type, only C has been changed, right?
Mm-hmm.
Okay, now if this is the double crossover, if you compare this one with the parental type, it's actually the middle gene will be changed, right?
Mm-hmm.
Does that make sense?
Yes.
Okay, now let's go back to that problem. Okay, so first we need to know, based on this data, this will be parental type, right? Because the numbers are too large, right?
Right. Yeah.
Yeah, this two smallest numbers represent the double crossover, right?
Mm-hmm.
Okay, now if you compare this one with this one, so which gene has been changed?
The CN.
Yes, so this gives us the conclusion CN must be standing in the middle.
Okay. So we will only see like one gene that's switched over for that one.
Yes, so yeah, so this is the way we can figure out the draft of the map. So which one is in the middle? So if you compare this one with this one, the same. So if you compare this one with this one, give you the same answer, right? Because these two are identical, right? And only
Mm-hmm.
this one has been changed.
Okay, got it. Thank you.
Sure. Okay. All right. So this is very important. And definitely, I'm going to give you a very similar question like this in your midterm. So yeah, make sure you understand the entire process, how we studied data from a 3-point test cross, and how we use the strategy I just showed you to figure out the draft map. So which gene of the three is standing in the middle, and then after that you need to calculate this map distance to derive the map like this. So you need to show me the order and also show me the distance here. And okay, so this is important. And also when you got all these numbers, so you need to know how to calculate the interference coefficient to let me know if in this region the crossing overs are independent from each other or not. Okay. So, all right. So we just in this 3-point test cross, we actually gave you, I actually gave you the example of genes, real genes, but in reality, if we do not, which gene is responsible, if we do not know, I mean, if we didn't know which gene is responsible for the traits, so which means if we want to study a new traits, but we do not know the gene, so what we can do is like we can leverage the molecular markers. So the markers the DNA markers in the genome to help us to find the DNA that is associated, DNA region that is associated with the trait of interest. And then we will zoom in this DNA region to dig out the real genes that are responsible for the traits. So let me repeat this, because in reality, if a trait is a new trait, not well studied, we don't know which gene is responsible for that trait. Then how do we do? So what we do is like we can leverage the DNA markers on the genome and use that to find an association between that marker and the traits. So once we identify that marker, we can look into the DNA region near that marker to identify those genes. All right. So the markers, actually DNA markers represents different forms of the variation at the molecular level. There are many types of DNA variants. So for example, the mostly commonly used one is the SNP, single nucleotide variant, which means like if you look at the genome in a population, so there's a single nucleotide change among the population between different people or different individuals in an organism. So that's the most common DNA variant. And also you can see, so there are some like DNA insertion or deletion, the entire block substitution, the inversion variant, or the copy number variant. So copy number means like, for example, there's a very special small sequence of DNA. And in one individual, you're gonna see just one copy here. But in another individual, you're gonna see there are two repeats here. Identical region, but for this individual, you're gonna see more than one copies. All right. So this is the forms of the DNA variants. So the markers, this variants can help us, as I mentioned, we can leverage these markers, DNA variants, to help us to identify genes that are associated or that are responsible for the traits, variability of a trait of interest. Okay, so in most of the situation, the marker, the DNA marker itself is not related to the phenotypic effects. For example, like the disease, the marker itself is not. But if the marker, let's say, if this is a marker, the SNP, the single nucleotide variant, if this SNP is very close to a gene, so suppose this is the diseased allele, this is the gene, and this allele, the mutant allele is responsible for the disorder of interest. But we do not know, suppose we do not know this gene is responsible for the trait, right? But if we can identify this marker is associated with the disease, then we can look into this genomic region, which can help us to identify this gene. Why is that? Because this marker, this single nucleotide variant is tightly linked with this gene. Now we know we should understand what does the tightly linked mean, right? Because the distance between this marker and the gene is very short, very small, such that the chance that any crossing over occurring between this marker and the gene is very tiny. The chance is very, very small, extremely small. So so in a population, this marker and this diseased allele tend to be inherited together. So if this is the case, then we can use this marker to help us to identify these genes. So this is what we're gonna learn now. All right, so marker, the single nucleotide marker is a very good tool for us to use in genetics. Why is that? because the markers. So because now we have very advanced sequencing technologies, and we can also use a lot of like molecular tools to help us to phenotyping these markers. So for example, if this marker has like either the homozygous locus or another homozygous locus or heterozygous locus. So what we see if we ran a gel for this DNA for different individuals carrying different genotypes here, so this homozygous genotype will give you a band like this, because and this homozygous will give you a band like this because they have different molecule weight. So if you run the gel, you're gonna see two different phenotypes, right? So it's easy. However, if you run the gel for the heterozygous individual, you will have both bands, which means okay, so this DNA marker is actually called the codominance. So it's different from what we have learned before. So when we talk about two alleles, one is dominant, the other one is recessive, which means like the big R, small r will give you identical phenotype as the big R, right? But in this case, if you look at the phenotype, this and this, this phenotype will be different from this phenotype, which means like the heterozygous individual will give you different phenotypes than the homozygous, or both allele will demonstrate that specific phenotype. So in this case, we're gonna say this locus is a codominance. So most of the marker has this feature, so it's very nice. Then we can easily phenotype them and use them to help us to do the genetic dissection. Okay, all right, this is the summary for the single nucleotide polymorphism. So we use a SNP to represent, so we have a short form for this, SNP, and this is the most commonly used DNA markers for genetic study, association study. All right, so SNPs can occur in any DNA sequences in the genome. So for example, the SNP can occur within a gene. So this is a gene, let's say this is a gene, the sequence of the gene, and the SNP could be within gene, or this is another gene. The SNP can be located in the intergenic region. So for example, the SNP can be situated here. So this is in between genes, right? And okay, so in human, in ourselves, there are about like there's about like one SNP per this many bases, which means there are many SNPs markers in our genomes, which is good because this many SNPs variant can be used as a very powerful tool to help us to identify genes of interest. Right? So for example, in every single chromosome, there might be this many 100,000 SNPs. So it's very densely distributed, so which is good. So as I mentioned, we can use them. So this means like everywhere in the genome, in our genome, there are some like useful SNP markers. So those are tools that we can leverage for the genetic dissection. All right. So next important concept is haplotype. Haplotype means the genotypes or the alleles that are inherited together. So if we talk about any individual, so for example, for a human being, so we have two chromosomes, right? Let's talk about chromosomes. So we have two chromosomes. One chromosome is from dad, the other chromosome is from your mom, and so the deadest chromosome has a specific combination of the alleles for all the genes. That's the entire haplotype, one haplotype for an individual. So the same, we have an entire set of alleles for all the genes from mom. So that's another haplotype for individual. However, when we talk about haplotype in a population, not an individual, so this haplotype, so when we talk about the haplotypes in a population, those haplotypes tend to be shorter, much shorter than a chromosome, right? Because we're talking about a population. And the haplotypes in a population tend to be inherited together quite frequently because they are rarely broken up by a crossover. So usually like a haplotype in a population is about like this length of this length, the 50 kb, K base pair. All right. So if you look at the entire length of a chromosome, the whole chromosome is about this long. All right. So the longer the chromosome, there's more chance for crossing over occurring, right? During the meiosis. So we have learned this. So if the haplotype is very short, so the chance to have crossing over within this haplotype is very low. Okay, is very low. So this is the reason why. So even in the population, entire population, after generations after generation, so you still see the intact haplotype because the haplotype is short, and it is very rarely to break up to chop up this haplotype by crossing over during the meiosis. And haplotypes eventually will decay. Decay means like randomized due to the recombination, due to the crossing over. Yeah. However, this process may take many years, many generations. So that's why still, yeah, when we look into our human beings, so we're gonna see like many intact haplotypes because the decay of those haplotypes, yeah, takes time. All right. So this is an example. So we can leverage the haplotype map in the mitochondrial DNA to study the human history, the evolution history in human, or the migration history in human. This is something related to the topic we covered before, right? The maternal inheritance pattern. So this is a trace back using the haplotype information from maternal line, from your mom and mom's mom. So yeah, because this is mitochondrial DNA. All right. Also, the same for the Y chromosome haplotype. So this is from the paternal line. So we can trace back to study the human history. And there's a very famous project called the 1000 Human Genome Project. So in this project, actually, scientists have genotyped more than 1,000 humans across on this planet, and by studying this genetic background and haplotype information, so we can derive the human history or relationship between different races. Yeah. So this is another example. Okay, so linkage disequilibrium, so LD, in short, LD. So this is another important term here. Okay, I believe everyone is familiar with this diagram, right? This is a classical Mendelian experiment based on the pea plants. All right, so this is different, right? Because when we talk about this diagram, we were talking about two genes that are independently assorted, which means these two genes are unlinked. They are on two different chromosomes, right? So if this is true, if they are unlinked, when we do the dihybrid cross, we're gonna get 4 categories of the phenotypes, and they're gonna follow this ratio, right? 9:3 to 3:1 ratio. All right. So this is totally random, which means these 2 genes are randomly assorted, and they are unlinked. All right. So now the LD is totally opposite. Is totally opposite. If these 2 genes are not random, if these 2 genes are not independently assorted, then what does that mean? That means these 2 genes are linked, right? If these 2 genes are linked, we say these 2 genes are in LD, linkage disequilibrium, or non-random association. All right, of the alleles at different loci. Okay, so I very much like this diagram, because this diagram gives us a very clear information about what we have learned so far. All right, so this is the starting point, right? So we start from here. It's very easy. Monohybrid test cross. Let's say this is monohybrid, and if we cross this with the double, the homozygous recessive, so this is called the monohybrid test cross, right? This gives you what? 1:1 ratio, right? Phenotypes. So we should be expert now. So if we talk about the monohybrid test cross, this will give you 1:1 ratio. So this is 1:1 ratio. The red and the black, right? All right. Now, if we do the monohybrid solved, what is that? So that is this, right? Solved means this, right? This gives you the classical ratio of 3:1. All right, so this is 3:1, right? Okay. All right, so this is 1 gene, so we don't worry about independent assortment. Now let's move to a little bit more complicated situation. So here, if we talk about 2 genes, dihybrid test cross, what is that? What is, let's say, okay, yellow, right? Okay, this is the test cross. This gives you 1:1:1:1, right? Okay, if we so remember, this represents the independent assortment. So this 1:1:1:1 only applies to the independent assortment, unlinked genes, not so not in LD, right? Okay. Now, still, if we talk about the unlinked genes, independent assortment, but in the dihybrid solved, this gives us this classical ratio here. Okay. All right. Let's look at the linked situation. So this is different. So if we talk about the dihybrid test cross, but linked, this will give you what? Also will give you 4 categories phenotypes, right? 2 are parental type, and the 2 are recombinant, right? So because of the linkage, you're not gonna see 1:1:1 ratio anymore. You're gonna see 2 categories have relatively much more progeny. So that's the parental type, and you're gonna see another 2 category which has much less progeny. That's the recombinant, right? So this is for the linked gene. So if you see the difference between here and here, then you can immediately make a conclusion. This is independent assortment, unlinked gene, and this is the linked gene, right? The same thing for here. So if we talk about the trihybrid test cross, but if the 3 genes are independently assorted, all 3 genes are on different chromosomes. Then we're going to see 1 to 1 to 1 to 1 to 1. So this is the ratio. But if we think about the trihybrid test cross. So if 3 genes are all linked, then we're going to see still 8 categories here, but only 2 are parental type. The biggest, these 2 will be parental. And we have like the other 4 categories represented a single crossing over. And finally, we have 2 categories represented the double crossing over. So that's the double crossing over. Okay. I hope this is clear. I very much like this diagram because it's very clear to show the difference between the independent assortment and the dependent assortment, the linked genes. Okay. All right. So let's look at how we can use the haplotype to help us to identify unknown genes. Suppose we're talking about a disease, all right? So this is the initial individual. So many years ago, there's one individual. So there's a mutation occurring in this gene, and this gene caused the disease. Okay, disorder. Let's say. Okay. All right. And in this individual, so there are like 7 markers here: 1, 2, 3, 4, 5, 6, and 7. All right. And this disease, the genes will be passed on from generation to generation, right? Okay. So however, because this marker is far removed from genes, right? So does the gene, the marker number 7. So during the generation, so generations after generation, when this diseased allele is being passed to next generation and next, so the crossing over could occur during the meiosis between any of these markers and this gene, right? Because the marker number 1 is so far removed from this gene, so it's easily that crossing over can occur between marker 1 and the genes, right? So you're gonna see change. So the random association between the marker 1 and this gene, and the same for the marker 7, and also for the 3, for the 4, and for the 3, for the 6, for the 2. So for many years, so after many years, and after many generations, what you can see in the population, so this gene has been, this disease allele has been dispersed in the entire population. So if you look at the sample from the whole population, so you're gonna see many different types, right? But you only see that this gene is tightly linked with the marker 4 and 5. Why? Because the marker 4 and 5 are so close to this diseased allele, so it's very rare to see crossing over occurring between gene 4 with this gene, with marker 4 with this gene, and the marker 5 with this gene. So in the whole population, the marker 4 and the marker 5 are always inherited together with this diseased allele. So we call this the haplotype. But because we do not know this diseased gene, right? We can only see this marker 4 and 5. Then when we analyze these markers, we can find the association, the significant association between the haplotype of marker 4 and marker 5 with the disease. Then we knew immediately, like, this gene should be close to these 2 markers. We can look into the DNA region containing these 2 markers, and then we can identify this diseased genes. This is how we use markers, haplotypes, to study a whole population to help us identify the genes of interest.
Professor?
Yeah.
So I don't, like, I know that if you, so for 4 and 5, it will be like preserved because it's near the mutation, but why does that happen? Because like what does a mutation being there have to do with how it gets?
Oh,
carried over?
the mutation itself is not related to the linkage between 4 and 5 and the gene, right? So let's say that this pink region represents the gene. So initially, without this mutation, this gene is functional and individual will show the normal phenotypes. However, many years ago, there's one individual for some reason, like maybe like a radiation or a chemical situation. So if or just random like mutation, so occurring in this individual, and the mutation and this mutation, suppose this mutation actually destroying the function of the gene, and this individual becomes affected by the disease. Okay, so this is a situation. The mutation only affected the gene function, not affected the linkage between this markers and the gene. Okay.
Right. So why do we see them carried over then?
Yes. Okay. So for example, this individual had a kid. So if there's no crossover, this individual will pass the entire this region to the kid, right? Okay. Now the kid carry the same thing, and the kid will be affected by the disease. Is that right? This is a dominant one, let's say. Okay. Now, when the kid also had a kid, so maybe there's a crossover occurring here. Okay. Then what happened? That means like probably these two markers will be changed, right? But this region will remain the same. Okay, so this is the first generation, and the next generation. So that the grandson carries this region, right? But okay, so if that grandson has another kid, so probably in that region, in that generation, so a crossing over occurring here, then these two markers will be changed. So these three markers will remain unchanged, right? Yeah. So you just imagine, keep going. So because those regions are those regions are far removed from the gene, so they have better chance to be changed. Only this 4 and 5, because they are tightly linked with this gene, the region between 4 and this gene and the 5 and the gene are very short. So it's very unlikely to have any crossing over occurring between them. That's why even after generations and the generations, this region will be reserved.
Okay, so that kind of has to do with like the fact that non-beneficial mutations will be selected out of a population, which means that 4 and 5, like, does that have anything to do with it? Like the reason that it doesn't cross over and
yeah, the reason why they didn't cross over is not related to the disease itself. It's because the distance. Because when we talk about like the region, the map region, so if the region is long, then you're gonna see more crossing over in this region, right? If this region is shorter, then you're gonna see less frequency of crossing over in this region. So this non-crossing over between them is due to the short distance between them. They are tightly linked on the chromosome. Does that make sense?
Yes, thank you.
Yeah, sure. So that's why. So if we can identify some markers that are associated with the disease, then we believe that the affecting genes should be around that marker. Yeah, that's the reasoning behind. All right. Okay. All right, so besides the SNPs, that's the single nucleotide polymorphism, right? So we have some other useful markers as well. So we call the simple sequence length polymorphism. So for example, here, so one square represents a specific sequence. So if we put 2 here, means like there's 2 repeats, and the 3 repeats, and the 4 repeats. Different people may carry different number of the repeats. And if the repeats numbers are different, and you can imagine the molecule weight should be different, right? So if you run the gel, so you're going to see different bands. So this is a very useful DNA markers that we can use for genetics study. So these DNA markers are also called a fingerprint. So we can use these markers in the forensics or for the parental test or the pedigree analysis. So based on the pedigree, so for example, in the parental test, you can see which child is the biological child for this marriage. So for example, this is the biological child from this marriage, and this is only the child from mom, not dad, and this is the biological child from this marriage, and this is a son adopted, totally adopted. Yeah, so there's no shared genetic information or the DNA markers, fingerprint between this adopted kid and their parents. All right, so this is very useful. So this is another example. So we can use the markers, DNA markers with different number of repeats and the different molecule weights to help us to study a pedigree to identify the genes that are responsible for this disease. So this, for example, this is a rare disease, and this rare disease only occurs in a family, not popular one. That's why we study the pedigree. So for example, this is the parent, the female parent is affected by the disease. This is a capital P, so dominant, and the mom has been affected. And so actually she is a heterozygous, so one big P and one small P. And the daddy is a normal one. So that's why he has homozygous little p and little p, all right? And because this gene is tightly linked with the marker here, so we can use this marker to help us to identify this gene, right? So this is what we just learned. Okay, if you look at the DNA, so you're gonna see each chromosome has a different marker because different repeats numbers here. All right, now if we study this pedigree, mom is affected, daddy is normal, and the children from this marriage, you're gonna see we have a bunch of like affected kids and a bunch of unaffected kids. So if we run the gel for their DNA, you're gonna see immediately identify this marker. So because all the affected individuals are associated with this marker, right? So this is the unique markers. And then what happened? So based on this, you can immediately identify. So we have many markers, but only this markers gives us the association between the marker and the disease phenotype. Then this can help us to identify this gene. This is an application of the DNA markers here. And the marker itself can be mapped because 3-point test cross, we try to derive the map for 3 genes, right? And so this is the same for the markers as well. So if we want to derive the map, genetic map for 3 markers, DNA markers, we can do the same thing. And this is how we can create the draft of the genome, each chromosome as well. So for example, we have many markers on a chromosome, then we can use the 3-point test cross to help us to derive the map for any 3 markers. And if we use this information, we can derive a longer map. So we can map 4, and imagine we can also extend this map by including more markers in the map. And if we put the small chromosomal region together, and we can develop a continuous linkage map called the contigs. So this is another genetic term, useful term. Okay, so so finally, I'm gonna give you an example. So this is a very good example. It's also from a textbook. So it's chapter 4, the solve the problem 1. It's very interesting. So here we are talking about a gene. Let's see. Okay, a dominant gene that is linked with the gene which determines your blood type. So actually, this is the gene determining people's blood type. So we know in humans that we have like A type, B type, AB type, and O type. Okay, so this means like, so if this is the case, so we should know, okay, this allele and this allele are the co-dominant, because when the heterozygous individual carries both allele, you're going to see both phenotypes, right? So that's the co-dominant, right? And in this special disease called the dominant misshapen nails syndrome, all right, in this family, and so it happens that this disease allele, the capital N, which is the dominant disease allele, is linked with the IB here in this female parent. All right, now look at the father, the grandfather and the grandmother here. So grandfather is actually the little I, just like a test cross, right? This is the homozygous recessive, and so we do the test cross like this, and so they have many kids here. Many of them are affected. For example, here, because they carry this disease allele, because the linkage between this IB with the disease allele, right? But as you can see, some exceptions in their kids. So for example, this one. So this one does not has the IB, but still is affected by the disease. And so, oops, no, not this one. And this one is very interesting. This one has the IB allele, but it is unaffected by the disease. Yeah, the same one for this one. So what can I explain for this? This can be explained for the recombination between these two genes, right? So if you think about this, you can make the conclusion like the disease seems to be dominant. Of course, it is dominant, and there's a linkage between this disease allele and this IB allele for the blood type genes. Okay, now, so how can we explain this individual and this individual and this individual exceptional because of the crossing over, right? Because if we do the test cross, so this parent, affected parent, made with this double recessive parent, and then majority of this progeny will be parental type, but only small proportion of the progeny showing the non-parental type, which is the recombinants, because of the crossing over occurring here, right? Right. And the problem, this question asks you to calculate the frequency of this cross. All right. So based on this diagram, so you can easily figure out the recombination frequency is about this. So this is a very interesting problem. So I hope, like, I don't think I have time to go to the details about this, but this is a very, very interesting and useful example to help us understand how we can use the linkage, the LD, the non-random assortment to help us to diagnose the diseased allele from a pedigree. All right. So I think I'm gonna stop here. So I'm gonna leave like 10 minutes for questions. So in case you have questions. Any questions for today? So yeah, thank you. So I saw a question in the chat box. Let me see. Do we have full midterm results? So probably yeah. Still, so you're still waiting. So yeah, we have too many students. So I think yeah, we're almost done with the grading. So probably tomorrow discussion, TA will get the results back to you. Your grade. Yeah, I hope so.
Hi, Dr. Jia.
Yeah.
Actually, I finished the midterm grading, and I
nice.
believe you should find your grading in the grade center.
Oh, okay, great. Thank you. So I hope everyone heard that. So Lei has already graded all the midterms, so you should be able to find out your grade from the I learned, yeah, the grade center. Yeah. All right. Thank you, Lei. No, I will not make any change at this moment. So I just for the midterm quiz, I will not make any changes for now. But finally, so after I got the points for the final exam, I'm gonna add up all the numbers together, and then yeah, that's the time I'm gonna make some decision to do the curving or stuff to yeah to give the final letter grade to every student. All right. If you do not have questions, thank you all, and see you on Thursday. Yeah. Good luck. Tomorrow is the quiz. All right. Bye-bye.